Hey everyone, I'm in a bit of a rush, so I'm gonna move quickly through everything. I definitely would look at this as more of a review, and I would use REM's lectures and Boards and Beyond, and there's a lot of great YouTube content to build a good foundation. And I created these last year. And so if I had more time, there's ideally a couple of things I'd like to modify. So some things might be a little bit of an MCAT refresher, a little bit, some of the stuff might be slightly misaligned with what you'll expect on the MBMEs. But all in all, I think this is a solid review, and certainly the later MFM biochem reviews get in more proper alignment with what you can expect from like the MBME step one material. And whenever in doubt, you should always just look at first aid as your Bible, that you can just make sure. And if something doesn't show up in first aid, it's probably not very high yield. And again, you'll see some of that with this lecture. So without further ado, let's get started. This is everything that we're going to be talking about in this lecture. It's about 120, 130 slides. So again, Boards and Beyond is great. First Aid is great. YouTube is great. REM is wonderful. I personally don't like Anki, but lots of people swear by it. Definitely just Googling things whenever you're unsure of something. That is an essential skill. Okay, so yeah, the OSCEs don't. A lot of this is just me kind of waving you guys off. Don't overthink it. It's not as bad as it seems. The OSCE, just look at it as a quiz. It's a fun time to collaborate with your colleagues. I wouldn't get so freaked out about it. The writing assignments, 3 to 8 hours, probably too much. I think if you spend 2 to 3 hours, and you try hard, you do a little bit of research, it'll be fine. Keep in mind that the OSCEs and the writing assignments really aren't that high in terms of your percentage. It depends if you want to be an MFM TA or not, in which case maybe you want every point that you can get. Okay, so the amino acids, a big refresher from the MCAT material. So we have our nonpolars, our positive charged histidine, lysine, and arginine. I would remember that these have a slightly higher PI, so they're going to be more positive than histidine, which hovers around like the physiological pH. I think their PK is, the PI is about 6, so that it can be easily protonated and deprotonated. If we're going through superlatives for everything, glycine's very small R group, means that it's very squishy. It's going to be good at turns. Cysteine can form disulfide bridges. Proline, because the R group loops back on the nitrogen, it lacks a hydrogen to participate in hydrogen bonding, so you won't see it in secondary structures and alpha sheets and alpha helices and beta sheets. Methionine, AUG is the first start codon. I would really just think of tryptophan, phenylalanine, and tyrosine actually belonging in the group together. These large aromatic molecules, very bulky, and when we're trying to detect how much protein we have, we can use the UV absorption around like 280 nanometers as a way of tracking the concentration and abundance of a particular protein sample. Serine, threonine, and to some extent tyrosine can be phosphorylated, so that's just going to switch a protein on or off. Often we do genetic engineering experiments where we might switch out a serine for like an alanine, and now we can't phosphorylate it. So if that phosphorylation event turned it on, it's permanently off. If that phosphorylation event turned it off, now it's permanently on. The opposite of that would be putting in an aspartate or glutamate, which is a phosphomimic. The negative charge of the aspartate in the glutamate mimics the phosphate. So now imagine that a phosphorylation turns something on, and a missense mutation turns that serine into an aspartate. Now it's going to be permanently on because the aspartate looks like a phosphate, and in this situation, that was a permanent on switch. And I'm trying to think what else. That's probably good. You really, this is probably more than you need, but it's just good stuff to know. Good background material. This is a little bit more aligned with the MBME right here, and stuff that you'll see in subsequent, not only MFM lectures, but also in subsequent classes. Phenylalanine becomes tyrosine, becomes DOPA, like L-DOPA for Parkinson's patients. They lack dopamine because the substantia nigra has been destroyed. And I would also know that as downstream of this, we have the catecholamines, norepinephrine, and epinephrine. I know this seems like a lot, but I really think all of this will come back again. A little tie-in here. I mean, if you think about the basal ganglia of the brain, we have the substantia nigra, which produces dopamine, and if we really just track the word substantia nigra, that's black substance in Latin. Why is it black? Well, it's because it has a lot of melanin. DOPA is a precursor for dopamine, which the substantia nigra uses to help coordinate motor function in the basal ganglia, but it also converts to neuromelanin, which is dark. So that's a little bit of a tie in there. Tryptophan, I would know that it becomes serotonin. it's a little silly and funny, but some people, and I was in Amsterdam this summer, and I guess they manufacture ecstasy there, and some people apparently it depletes all of your serotonin, and so what some people do is they'll have a lot of turkey or milk the next day because this is very high in tryptophan, and supposedly the tryptophan replenishes your serotonin by generating it. Similarly, a lot of people say that after Thanksgiving, you get really sleepy because you're eating a lot of turkey, and turkey has a lot of tryptophan, and tryptophan becomes serotonin, which becomes melatonin, which makes you tired. Both of these are probably not true. It's probably, if you think of the second one, you're up all night cooking, and then you're drinking lots of beer and eating lots of food. That's going to make anybody sleepy. So, but that's maybe just a nice way to remember it. Histidine, hist, and histamine, hist go together. Glycine makes heme and hemoglobin. Glutamate, I just think of the G's. Glutamate makes GABA, the inhibitory neurotransmitter. It also makes glutathione, which is an antioxidant. Arginine becomes a lot of things, becomes creatinine, which we have a lot in our muscles. Urea, which is a waste byproduct for the ammonia that we generate from the nitrogen, which where urine gets its name because we have a lot of urea excreted there, and nitric oxide. Cool. So there's maybe 4 different ways we can conceptualize a protein. The primary structure is literally just the sequence of amino acids. A lysine follows a valine, follows a tryptophan. You can just think of this as like the arrangement of letters chronologically in a word, right? Elephant, E-L-E-P-H, right? Just what letter follows the other one. That is the primary structure. Peptide bonds link these together. The secondary structure, we have alpha helices. This is going to be hydrogen bonding between the backbone, the nitrogen's hydrogen and the carbonyl C double bond O. Same thing for beta sheets. Those are the 2 main ones. We do have turns. We do have unstructured regions, and again, you can imagine that something like proline is unlikely to be found in these because the looping back on the nitrogen depletes it of a hydrogen, and it's incapable of doing hydrogen bonding. Tertiary structure is, you can just think of the secondary structures folded in on themselves, but specifically this is going to be interactions between R groups within the protein. And then the quaternary structure is really just interactions between R groups, but between different polypeptide chains, right? The classic example is hemoglobin. Hemoglobin is made up of 2 alpha chains and 2 beta chains. So that means that every hemoglobin protein has 4 N terminals and 4 C terminals. Peptide bonds. So anytime we link up monomers, it's a dehydration reaction. We're taking out this OH and that H, so that's effectively a water that's being depleted. This brings them together, and if we see that there's a lone pair here, we can imagine that there is resonance to kick that up. So there's going to be double bond characteristics here. And the double bond is important. You can rotate around single bonds. You cannot rotate around double bonds. And so that is going to contribute significantly to protein shapes. I would ignore something like psi and phi angles. Those are important. Ramachandran plot, probably not that important either. Being able to just recognize like a general polypeptide, right? Here's one amino acid, and here's another amino acid, and here's another amino acid, and here's our R group, so that would be a glycine, and this would be an alanine, and then we have glutamate, lysine, et cetera, et cetera, et cetera. The N terminus is made first, and then the C terminus trails is the last thing that is going to be popped out of the ribosome during translation. And again, we have these little dashed lines right on the peptide bonds, which again are going to be sp2, double bond characteristic, difficult to rotate. And we mentioned alpha helices and beta sheets. These are just the 2 most favorable structures, just the way that all of the sterics and whatnot worked out. These are just the arrangements that you'll see. These are the favorable bond angles. It's going to be this, or unstructured. R group interactions are going to play a role in the tertiary interactions. We can have electrostatic interactions, also known as the salt bridges. So you could think of like an aspartate and a lysine, or glutamate and a lysine, or you can think of van der Waals forces. Also, you can think of as hydrophobic interactions, or London dispersion forces, or instantaneous dipole-induced dipole. A lot of these names are kind of interchangeable, but this is a bunch of non-polar things just kind of aggregating together. Of course, hydrogen bonding, you can have dipole-dipole, but these are just lots and more. You can also have instead of non-covalent interactions, intermolecular interactions. You can have intramolecular interactions. So the example here, really the only example would be 2 cysteines forming a disulfide bridge. All right, so looking at pK, charge, and titration and whatnot, the every amino acid is going to have two ionizable groups, right? The C terminus and the N terminus, but only these 5 will really have a significant ionizable group in terms of the R group. And even histidine, notice it's pKa for the side chain is around 6, around physiological pH. So it's going to go, it's going to be deprotonated, protonated, deprotonated, protonated frequently, which is going to be the explanation for why it's found in a lot of the enzymatic sites of a protein. But lysine and arginine are going to be the very positive ones, right? If you're thinking of a histone protein, what do histones do? They bind to DNA. They help organize it, right? Heterochromatin, euchromatin. Well, if a protein wants to bind DNA and DNA is negatively charged because of the phosphodiester backbone, what do we expect to see an abundance of in histones? Probably lysines and arginines. And then aspartate and glutamates are just going to be your negatives for the most part. So the C terminus is a pKa of 2. Again, we're getting a little bit in the weeds. Like, this is technically fair game, but I think it's low yield. That's why I'm definitely rushing past something that is enormously complex. So hopefully you guys remember a fair amount from your undergrad days to help this go smoother. But the pKa, you can think of it as, if the pH is equal to 2, 50% of this is in the form we see now. It's a carboxylic acid, COOH. 50% if the pH is 2. If at the same time, 50% of it will be in COO minus, meaning that we've lost this hydrogen here. So at a pH of 1, you can imagine that it's 100% COOH at a pH of 1 and a pH of 0 or a pH of negative 1. Yes, you can have negative pHs. And then at a pH of 3, 4, 5, 6, 7, anything above, now it's going to be 100% COO minus. If we're looking at this, what are we seeing? We're seeing that at a pH of 10, it's going to be 50% pronated, 50% depronated. Here at a pH of 4, 50% pronated, 50% depronated. So what's the story if I have a pH of 4? If it's a pH of 4, we can imagine that this is going to be 100% minus. And of course, it's not 100%. It's like 99 point. We could calculate it, but you don't need to. You won't be expected to for medical school. So 100% minus. This is going to be 50-50 at a pH of 4. And this one it hasn't lost its hydrogen at all. So this is going to be 100%, just we can say pronated. And just a shorthand of this is that the lower the pKa, the more acidic. So here we have the best acid, the second best acid, and here we have the worst acid, that is to say the best base. Looking at a titration curve for this, so right on the x-axis is, imagine that we have a beaker and it's filled with some acid HA, and it might even be H2A, right? We have 2 possible hydrogens we could lose, and we're throwing in OH minus, we're throwing in a base. And so this x-axis is saying, hey, what's going to happen the more OH minus, the more base that we throw in? Well, you can imagine, just as a general rule, if the x-axis is the concentration or the amount of moles of OH we're adding, and the y-axis is the pH, well, as you add OH, your pH is going to go higher. You're going to be more basic. And that's the general trend that we do see, right? This does go, this does have sort of a positive slope, but it has these interesting, tricky curves. And we could spend a whole 45 minutes on this. It's so not important and relevant. So if you're freaking out that this is a lot, you could probably not know any of this, and maybe you'll miss 1 or 2 questions, and maybe you won't miss any. This is not high yield. I'm just talking about it a little bit for fun, but at the same time, hopefully it kind of clicks. So if we imagine we start off at a pH of 1 here or 0.2, what are the ionizable groups for this glycine? It's just going to be the N terminus and the C terminus here. And the pK for this is about 2, and the pK for this is about 9. So at a pH of 0.2, it should be, this is going to be 100% pronated, and this is 100% pronated. And so everything is in the +1 state. And then what happens when I get the pH to about 2? Specifically 2.34, but let's just say 2. The pH at about 2, we're now saying that what's the more acidic one? It's the carboxylic acid. This should be about 50-50 at this point. And the 50-50 at this point, is showing that we're going to be right in here. We could say that 50, it's going to be NH3 positive COOH. This is going to be 50%. And right here we have NH3 positive and COO minus. This is what we have at around a pH of 2, pH of 2.3. And you can think of the charge here, if this is a charge of +1 and this is the plus and the minus cancel, this is neutral. If I have 50% of a +1 charge and 50% of a neutral charge, you could think of this as a 0.5 plus charge right here. We have an equal amount of both species, and if we go back to our Henderson-Hasselbalch formula from Gin Chem, pH equals pKa plus log A minus over HA. And you know what? If I have 50% and 50%, whatever those numbers are, it's going to be the log of like 0.2 over 0.2, or the log of, 10 to the -4 over 10 to the -4. The log of 1 is 0, so that cancels, and the pH is just equal to the pKa, which is why this works and matches up so nicely. So here we know that the pKa of the carboxylic acid and the C-terminus is 2, and so that corresponds with this buffer zone here. And the next buffer zone is going to be, we said, about 9, and so this is going to be about 9 or 9.6, it depends. This is going to be our next buffer zone where we have 50% of the NH3 plus and 50% of the NH2. But if you just kind of move up stepwise, right, you can think of this is +1, this is 0.5 plus, this is neutral, in other words 0, this is -0.5, and up here we have -1. So this is the worst case nightmare scenario problem. I would, if you know this, great, but if you just want a simpler understanding, so yeah, maybe no Henderson-Hasselbalch. There we go. We already said all that. If you want a simpler understanding, just know that the lower the pKa, the more acidic. And if the pKa is 2, that means 50% of the pronated version of that and the depronated version of that. So to low pH, we would have both versions of these pronated. At a medium pH, because this is the worst acid, it's still 100% pronated. And because this loses its hydrogen, we're going to have a point where we're going to have 50% O minus and 50% OH, but a little bit past that, now it's 100% O minus. This is a Zwitter ion where we have a plus and a minus kind of canceling each other out. And then at a high pH, we're now going to lose the worst acid, and that's depronated, and that's depronated. All right, we already talked about this. Histidine is hovering right around physiological pH, and so it's going to very frequently lose a hydrogen, gain hydrogen, lose a hydrogen, gain a hydrogen. And that dynamic nature is going to, an enzyme is dynamic. An enzyme is going from the unreacted form to the reacted form, unreacted form to the reacted form. So this is kind of a useful switch for that sometimes. Hemoglobin. All right, hemoglobin has quaternary structure. It's 2 alpha chains and 2 beta chains, at least the main hemoglobin. This is the only hemoglobin you probably learned in undergrad for the MCAT. There are other types of hemoglobin, which we'll talk about. Every one of these tertiary structures, an alpha chain or a beta chain, has a heme group. It's a cofactor, it's a prosthetic group. And at the center of that, we have this little iron molecule. And remember that glycine helps make the perforin ring, which is effectively makes up the heme molecule, which is, again, necessary to help us grab onto oxygen, which is the whole point here, right? Red blood cells are just bags of hemoglobin, right? They don't even have nuclei. They don't have mitochondria, because we just want, it's like in the movie Pearl Harbor where they evacuate everything from the plane just so that they can squeeze as much bombs as they can into the plane, because that's the mission, right? Same thing here. No mitochondria, no nucleus, just get everything out. We just want as much hemoglobin as possible, and then it just delivers oxygen. So here we have we know this comes from glycine, this perforin ring. We have iron at the center, and the iron is going to be interacting with oxygen, binding onto it. More importantly here is knowing these different types of hemoglobin. So the main one is hemoglobin A. This is like 97% of all adult hemoglobins, and it's 2 alpha chains and 2 beta chains. You do have like 2 or 3% of A2, which is alpha chains and delta chains. Mildly important, I wouldn't know this. Fetal hemoglobin, even more important. So during, as the name implies, fetal hemoglobin, during your first 6 months of life, you're producing actually a high amount of this. And it's 2 alpha chains and 2 gamma chains. So when in doubt, just remember that the alpha chains are in most things. It's the beta, delta, gamma that's going to vary from hemoglobin to hemoglobin. But 2 gamma chains, 2 alpha chains, we will come back to this. We can ignore the embryonic one, not so important. And we'll talk about these abnormal ones in a bit. You have something called H hemoglobin, which is 4 beta things together, tetramer, and the BART is 2 gamma. So we'll come back to this. As adults, we do have a tiny amount. We might have like 0.1% fetal hemoglobin. Fetal hemoglobin also has a slightly higher affinity for oxygen. And you can think the baby's a little parasite, right? It wants to steal this oxygen from mom. So, its needs are greater than mom's. Maybe it's more vulnerable. So it makes sense that it has like a stronger magnet for the oxygen than the maternal, adult hemoglobin. If we look at the myoglobin versus the hemoglobin, myoglobin is muscle. It's a tertiary structure. There's just one of them, right? Hemoglobin, though, is 4. So that is one difference. Another difference is that hemoglobin displays something called cooperativity as a result of the fact that it has 4 of them. So if you bind, it's just like this peer pressure effect. If I have 4 different sites, maybe this is my hemoglobin, there's no oxygen. It's kind of like, I always thought of a middle school dance. Nobody wants to be the first one on the dance floor. It's just kind of awkward, right? But the first oxygen to bind onto one of the heme groups, that's like the first couple going out and dancing. And that kind of makes things a little bit less awkward, so that the second couple is much more likely to follow, right? Maybe it takes 30 minutes for the first couple to go out to dance, and maybe it takes like 5 minutes for the next couple to come out to dance. The next oxygen binds more easily. And then it only takes a minute for the next oxygen to go to the dance, right? And then maybe it only takes like 2 seconds for the next one to go. And then offloading is going to be the same thing. It's difficult to be the first to offload, a little bit easier to be the second, and the most difficult to be the last. So that's cooperativity. It's this peer pressure effect, and Hill coefficient greater than 1, all of that fun stuff from the MCAT. And notice as well that there is a lower KD for myoglobin than hemoglobin. That is to say that myoglobin has a higher affinity for oxygen. There's a reoccurring theme in medicine and biology that the muscles are just assholes and they're selfish and they're going to hoard sugar, right? They hoard glycogen, they hoard oxygen, and they don't want to give it to the rest of the body. And on the other hand, the liver is often going to be this altruistic thing, constantly concerned for the welfare of the rest of the body. But so myoglobin hoards onto oxygen, it hoards onto glycogen. It doesn't, it's saving it for itself. And this is indicative, kind of like the fetal hemoglobin was like selfish and better at grabbing onto oxygen. Myoglobin is especially better. So myoglobin is single, it is not cooperative, and it is a higher affinity that is a lower KD. And we might call this like a hyperbolic curve versus like a sigmoidal curve. Hyperbolic myoglobin, sigmoidal hemoglobin, kind of like an S shape a little bit. Most of the oxygen in your body is actually bound to hemoglobin. So this is total blood oxygen, and this is the amount bound to hemoglobin. And notice that the values are really close, right? So 99.9% of the oxygen in your body bound to hemoglobin. Tiny amount is going to dissolve in the plasma. Oxygen really does not want to be free floating in the blood. So the incredible importance of hemoglobin to move oxygen around. Obviously, if you just think here, we're going to have more oxygen in our lungs where alveoli are, and your tissues is going to be lower. So in your lungs, you're going to have about 100% of your oxygen bound to hemoglobin. But when we go into the lower oxygen tissues, this Le Chatelier equilibrium dissociation dance means that a fair amount actually is going to leave. And it depends which tissues and how much you're exercising and things like that. But, showing here, we could lose maybe 25% of our oxygen when we go from the lungs to the tissue, right? And that's a good thing. We want the hemoglobin to let go of the oxygen so that we can, power aerobic respiration and whatnot, the final electron acceptor. Way I always remember this was, we'll ignore the carbon monoxide. Maybe we won't. Let's start with carbon monoxide. Carbon monoxide is just like oxygen, except that it has an even higher affinity for hemoglobin. Think about this. Oxygen is like this, right? And there's a natural polarization. There's an instantaneous dipole effect where, the electrons fluctuating might mean that at one moment, one of the sides is partially negative and the other side is partially positive, and that will be attracted to the iron. But it's going to be even more substantial if I'm looking at something like this, where this is because the oxygen is more electronegative than the carbon. It's going to be even more negative, and this is going to be a little bit positive. So the positive iron, 2 plus, 3 plus, whatever, 2 plus, is going to be even more attracted to the oxygen of a carbon monoxide. So that's like a useful way to remember, right? Just thinking about the actual underlying chemistry. So carbon monoxide has 50 to 200 times higher attraction, gravitational pull to the iron of a heme molecule in hemoglobin. All right, now that that's out of the way, let's talk about the more important things. Right shift, you could also think of it as a downshift, right? Right shift or downshift, however you want. We're losing affinity for our oxygen. And this is a good thing. Why? What situation would this come up, right? Let's think about temperature and H plus and really increased CO2. So increased CO2, which we'll see in a second, interacts with carbonic anhydrase. CO2 plus H2O makes H2CO3, carbonic acid, which dissociates to make bicarbonate and an H plus molecule. The point is, and you need to know this, CO2 is going to help make more H plus. So those go together. But so when am I going to have more CO2, more H plus, and a higher temperature? When I'm, let's say, exercising, or let's more fun. Let's say running from a bear. Right? From running from a bear for, it's been like 20 minutes. I'm just really, really cranking it. And my what's going on? My body's working really hard. My muscles are pumping very vigorously, generating a lot of CO2. Okay, CO2 is up. All right. And because I'm burning so much glucose, the aerobic respiration is in full sprint. We're processing so much glucose that we actually can't fit it all into aerobic respiration. Some goes to anaerobic respiration, which makes lactic acid. Lactic acid is an acid, which makes H plus. Additionally, like we just said, the CO2 is going to interact with carbonic anhydrase, make carbonic acid, and that's also an acid which increases H plus. And of course, if you're if you're doing this very exergonic reaction a lot, you're going to be creating a lot of heat, so your temperature is going to go up. Obviously, if you're running a lot, if you're exercising, you're going to get sweaty. So wouldn't it be great if a system evolved that when these three things are together, there's high CO2, high H plus, high temperature, we cause our hemoglobin to lose affinity for the oxygen? Why would we want it to lose affinity for the oxygen? Well, because I'm running from a bear. I need as much oxygen as I can get. What's going to be better? If my oxygen's sitting on a hemoglobin for a rainy day, or if my hemoglobin releases the oxygen, because this might be the last chance I ever get to use it, that is obviously better. So we want to communicate to the hemoglobin to, hey, loosen your affinity. We really need the oxygen here. Now is not the time to pinch pennies. Let it all go. And so these three things, this all kind of hopefully comes together a little bit. And the opposite process for decreased temperature, decreased H plus, and decreased CO2. 2,3-DPG, remember that this is an offshoot from 1,3-bis-phosphoglycerate, a intermediate in glycolysis, which you do a lot of in the red blood cells. There's a connection there. But 2,3-BPG is something that if you're, living, you're a Sherpa, living up in the mountains, your body naturally produces a lot more 2,3-DPG. And this is going to stabilize the tense conformation of hemoglobin, which just means that it's going to be more likely to offload oxygen, less likely to bind oxygen. So there's a kind of different mechanism there, but it's a similar right shift, downshift. All right, so we have carboxyhemoglobin where carbon monoxide binds instead of O2. And we just said that this is a super high affinity. So if you watch House of Cards, or you've probably heard of this, if you leave your car in the garage and close the doors and leave the car on, it's generating a lot of carbon monoxide, which you can breathe in and cause you to suffocate because it's binding to all of your hemoglobin. When a pulse oximeter, if anyone here is an EMT or just worked a little bit in the hospital, tells you your oxygen saturation. Ideally, it's high. Some people have emphysema or something, and it might be lower, but this is misleading. If someone comes in with a carbon monoxide, it looks like their oxygen saturation is very high, but actually, this thing is just seeing, is hemoglobin bound to anything at a heme group? It can't really differentiate very well between oxygen and carbon monoxide. So it is a potential false positive. If someone has carbon monoxide poisoning, you do a pulse ox and it looks fine. It's actually not fine. We can also have for various reasons, our iron further oxidized from the 2+ state to the 3+ state. And this is going to be kind of a complicated effect. So this is 3+. The actual, so these are the four, polypeptides of hemoglobin, the four globin chains in the hemes. This one is less able to bind oxygen, but it makes the other ones more tightly binding. And then the net effect of this is just you're going to get less oxygen out to your tissues. And this picture here is just showing that there's a very high affinity for carbon monoxide. To get oxygen half filled, you need a fair amount of oxygen, right? Let's say you need 25. But to get the hemoglobin half filled, if you just had carbon monoxide around, you'd only need like 2 or 3 units of CO, probably even less. All right, CO2 when it's pushed out from the tissues, hemoglobin or red blood cells carry hemoglobin and oxygen to the tissues, and then they take the waste, the CO2 from the tissues, bring it to the lungs, so you can exhale it, push it out. The CO2 is going to be pushed into the red blood cells, where we said earlier it's going to interact with carbonic anhydrase. You probably remember this from the MCAT. This is really important for the blood buffering system, which you'll have a whole thing about with the renal unit in the spring. This generates carbonic acid, H2CO3, which is not shown. That is going to immediately dissociate, because it's an acid, to bicarbonate and H+. The H+ can bind to the hemoglobin, and that's actually what changes the affinity of the hemoglobin to make it have a lower affinity for oxygen, so that we release it. Again, that makes sense. If you're exercising, you want this to happen. The bicarb, though, doesn't stay in the red blood cell. It's going to be pushed out. It goes out and it trades place with an antiport system with a chlorine. So you can imagine that if you were looking at the pre-capillary bed, you might see a lot of chlorine. And then the post-capillary bed, after we got a bunch of CO2 and had it converted to carbonic acid and bicarbonate, now that's going to switch place. So we would now see the chlorine concentration be decrease in the post-capillary bed compared to the pre-capillary bed. About 70% of your your carbon dioxide is actually transported through the blood as bicarb. I think that's really important to know. Bohr-Haldane effect, put simply, CO2 helps facilitate the offloading of oxygen from hemoglobin. And again, mainly it does that by CO2, carbonic anhydrase makes bicarbonate, and H+. H+ literally binds to the hemoglobin, changes its conformation to a conformation that's called tense, where it actually doesn't want to hold on to the oxygen. It spits out the oxygen. So Bohr effect, CO2 pushes O2 out. Haldane effect, the exact opposite. Oxygen comes and it's going to bind, and that's going to push the H+ out. And if we have more H+, it combines with the bicarb, which is going to generate more CO2. So they're really just opposite effects, more or less. So we mentioned earlier that most of the oxygen is bound to hemoglobin, like 98.5%. And carbon dioxide is bicarb, 60, 70, 80% or so. So most of the carbon dioxide is bicarb. We do have a little bit that actually directly binds to hemoglobin, and some is physically dissolved more than oxygen. We'll have a whole in-depth thing on this for the pulmonary unit in the spring. Thalassemia. So let's just focus on hemoglobin A, the main one. So 2 alpha and 2 beta. Hemoglobin A2, 2 alpha, 2 delta, and then hemoglobin F for the first 6 months of life. We have 2 alpha and 2 gamma. Remember that alphas are pretty much in everything. You have 2 beta globin genes, and you have 4 alpha globin genes. So this difference, this 1 to 2 stoichiometric ratio, is going to be important when we're thinking about the percentages in a second, looking at the thalassemias. Thalassemia really just means you have a defect in hemoglobin production. And it can happen in a variety of different ways for a variety of different reasons. But we're really going to focus on these numbers. So again, main one is hemoglobin A, 2 beta, 2 alpha, but we have 4 alpha globin genes, so we have more than we need a little bit. So anybody who's Mediterranean or Southeast Asian or African heritage, or genetics, descendants, whatever, is likely, much more likely to have a thalassemia than anybody else. So they might mention that in a question stand, in a vignette. They'll say, somebody from Greece comes over and they're anemic and they're pale, and I guess they might not tell you they're anemic, but they're tired all the time and they're pale, and you go, aha, this is a thalassemia. There's other diseases that would cause this, which we'll get to, but this is the one for right now. So alpha thalassemia, you have a defect in producing the alpha chain. Obviously normal is you have 4 functional alpha chains, you're good. If you have 1 broken alpha gene, so these alpha mutations, the red, that means we're not producing any alpha gene, none. But you know what? We have more than we need. We only need, in a hemoglobin A, we only need 2 alpha globin, proteins, but we have 4 normally, so we have way more than we need. So we kind of have like backups. We have like spares. So if you're just missing 1, you're probably fine. In fact, even if you're missing 2, we now call this, alpha thalassemia minor. We'll call this thalassemia trait often. So trait is the word I would know. Whether or not they're on different chromosomes or they're on the same chromosome, it doesn't really matter. You might have mild anemia. I would know that. All right, now it really gets difficult actually when you're missing 3. So we call this hemoglobin H disease. Now you're symptomatic. You're much more anemic, and hemolytic and microcytic anemia, and your red blood cells are going to burst, and your red blood cells are bursting and malformed and dysfunctional. And when you have all this crappy red blood cells because the hemoglobin inside it's precipitating and not doing the right thing, it makes the red blood cells get sick. And if you have a sick red blood cell, who takes care of the red blood cell? The spleen. That's really the spleen's job, is to eat up old red blood cells. Red blood cells have, they last about 120 days and then they're gone, right? But if you have alpha thalassemia, beta thalassemia, your hemoglobin sucks, or your red blood cells suck, so there's a much quicker turnover. And your spleen can only take so many red blood cells at a time, so it just swells up with all of this red blood cell garbage that it's trying to sort of metabolize, and then so you get splenomegaly. I definitely would know that. You might have extramedullary hematopoiesis, which is where your liver and your spleen are trying to actually, during, like, when you're an infant and during like embryogenic processes, you actually make a little bit of red blood. So hematopoiesis is like sort of the bone marrow is making blood cells, white blood cells, red blood cells. Normally your bone marrow does that. And if you're not making enough, then your spleen might, it's kind of awakens, right? Because during embryogenesis, it's making some stuff, but now that and the liver make it too. The skin can even do it, and you get this like bluish. They call it like blueberry skin because it's trying to produce all of this stuff. But I would say the main reason you're getting splenomegaly is because too many red blood cells are getting destroyed. You also can notice these targets. You get these target cells. I'll Google what a normal erythrocyte looks like, but this is a target cell. It literally looks like a target, hence the name. So that's a thalassemia issue. And you get these beta tetramers. So hemoglobin H is a beta tetramer. Why would this occur? Well, if we go back and we just think about the stoichiometry of this, if I don't have this and I don't have this and I don't have this, I'm gonna have more betas than alphas. So what's gonna happen is the betas are gonna go, I guess there's not enough alphas to pair with. Maybe let's pair with each other. And then the betas start coming together, beta, 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 and they form a tetramers. And those tetramers don't really work, and they start precipitating, and that causes some of the things that we see. So this is hemoglobin H is when you have a bunch of beta tetramers, and that might show that you have this more severe alpha thalassemia. All right, the fourth one is you have absolutely no alpha chains, incompatible with life. You're not going to be able to live if you don't have any alpha chains, right? Because hemoglobin A is the main one, and that's just no way around it. So in this case, you get hydrops fetalis. So this one here, you could be, I don't exactly know like the ages, but I don't know. Let's say you could be a kid. You could be like a 7-year-old kid with hemoglobin H disease. You might be very sickly, but you could have it. Bart's disease, the 4 missing, in the vignette, it's going to be a baby who was just born who's very sick and is dying. A hydrops fetalis is, so you have severe anemia because your hemoglobin doesn't work. You can't get oxygen around. And maybe you get a little bit around from the hemoglobin, I guess a little bit of maternal hemoglobin. So you can get a little teeny bit out, but you know what happens is, so to help that, because you're not getting enough out, the heart is going to pump extra hard to push more fluid around to try to pick up the slack to further oxygenate the tissues. And a recurring like motif in medicine and biology is that if you overstress an organ, it's just going to die, right? Like diabetes, if somebody is eating too much sugar and the insulin's working too hard, it just kind of craps out and the receptors stop responding. Here we're seeing that if the heart is pumping too hard, it's just going to crap out. You get heart failure, the heart dies, and if you don't have enough, heart's not pumping, think about the MCAT, think about capillary fluid exchanges, gas exchanges. If we don't have, there's osmotic pressure and hydrostatic pressure. The hydrostatic pressure is low because we're not pumping the heart very much. That means that we're not going to push out enough, and we're actually going to push more in from the interstitial fluid. This is, it's it's the opposite. From the plasma, we're not pushing enough out, and this is going to result in massive edema if you can't. Sorry. If you get heart failure, typically you get massive edema because of this hydrostatic osmotic like differential, and so the baby's gonna swell up. Basically, you don't really need to know all that. It's just like a fun, nice little connection to make. For the incompatible with life hydrops fetalis, I would know that this is Bart's disease, and this is a gamma tetramer. So why would you get a gamma tetramer here and not earlier? Remember that if we look at this, hemoglobin F is like 0.1%. So there's actually very little gamma. So even if I have a little bit of alpha, it's much more likely that, like, there's just not enough gammas interacting when I have one alpha around that, because it's already in such low abundance that I'm likely to see it precipitate out. If we have absolutely no alpha chains, then the gammas are eventually just going to accumulate, gamma, 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 and now we get the BART tetramer. Beta thalassemia is a little bit different, right? Alpha is like it's not functional. You actually could have a reduced functionality or a zero functionality. So there's a little bit of variability in there. I would just know that as a fact. I wouldn't memorize, this too specifically and significantly. Beta thalassemia minor. So you have one that's producing less or not producing at all, and this is usually silent. There's no clinical physical presentation or phenotype. Intermediate is where you have two mild ones, or mild expression and no expression, and then the major is going to be you have absolutely no beta. This gives you Cooley's anemia, very severe anemia. So anemia, they're going to be tired because they're not getting oxygen around, and if you don't have enough oxygen too, it's going to make you like a little bit pale, so they're going to have like a pallor, and but the Cooley's anemia here. It screws with like the development, and you're going to have skeletal malformations known as chipmunk facies, and you get something like this. So they'll mention something like that in the vignette likely. So why would there be a 6-ish month delay in symptoms, right? So someone could have beta thalassemia major or even the intermediate one, but you don't actually know until the baby hits 6 or 7 months. Well, remember that fetal hemoglobin is around for the first 6 months of life. And fetal hemoglobin so we said 0.1%, but again, that is 0.1% as an adult. During the first 6 months, it's actually a largish percent. And it is alpha gamma. It doesn't need beta. So you have a lot of fetal hemoglobin and it works fine. And for that reason, you shouldn't see any symptoms at 3 or 4 months. They love to ask it. That's really high yield. How does the medication hydroxyurea help? Hydroxyurea will stimulate, for whatever mechanism, I think it's unknown, stimulate the production of fetal hemoglobin in adults, which is kind of weird. We'll also use this for sickle cell, which will come up soon. This is just kind of a summary. Again, a lot of these things are going to lead to hepatosplenomegaly. So this, I would know, you might see some things with hard numbers, you might see things with up arrows, down arrows, and maybe I would just know the gist, right? I would know the big picture. Most of what we have, normal adults, very high A levels. We have a teeny amount of A2, which I'm really not going to talk about right now. You can kind of infer what's going to happen with the A2 levels if you just remember that there are 2 alpha chains and 2 delta chains. I think they say gamma here, but I'm pretty sure they're delta chains. Let me just double check on that. Yeah, they're 2 delta chains, so that was a mistype that you just saw. All right, so A2 is 2 alpha and 2 delta chains. But we're ignoring it. I don't think it's that important. Hemoglobin F, and then hemoglobin H, which is a beta tetramer, and the BART, which is a gamma tetramer. So if we have a lot of A, very little F, like 0.1%. You should have no beta tetramers, no gamma tetramers. All right, alpha thalassemia trait. Remember, this is you're missing like 2. It'd be kind of the same thing if you're just missing 1. Maybe your A goes down a teeny tiny percent, right? From like 97% to 96%, it won't be a significant impact. A2 and F should be affected about the same, right? Because they're also made using the alpha. And we shouldn't have any beta 4, gamma 4. Because remember, we have 4 alpha genes normally. So if you're missing 1 or 2, like it's really not a big deal. Again, 2 is obviously worse than 1, but the numbers should be more or less the same. Now, when it's alpha thalassemia H disease, you're missing 3. And what's weird is your HbA is actually still kind of high-ish to medium, but it's not very high. I don't know the numbers, but let's say it's 50 to 90%. Again, the point of these tests is not to ask you exactly what the percentage will be of the hemoglobin A if you have H disease. It's just to say, does it go up or down? And it's going to go down because we only have 1 alpha gene to make an alpha chain to pair up with the 2 beta gene chains. So that's going to go down. And we now have we have more betas than alphas. And so the betas are going to start to pair together. We get a little HBH. We don't get a lot of gamma tetramers because remember the fetal hemoglobin is only like 0.1%. So even though you might think that's like the same ratios as like the beta, it's just not much. The beta is much more significant, 97% hemoglobin A versus like the 0.1% in the fetal. So it's actually still quite low. Now in Bart's, you have absolutely nothing. Remember that the H disease in Bart's might have target cells. Bart's is going to be hydrops fetalis. Your heart's pumping really hard to compensate or try to compensate for the lack of oxygenation, leads to heart failure, leads to edema. You can't make any of this because it needs alpha. You have none. You can't make any of this. You can't make any A2. You have tons of beta things that are interacting. And this is the significant one here, is that the gamma tetramers are now going to start precipitating and coming together. And that's because, I know it's only 0.1%, but there's just literally nothing. So over time, that 0.1% the gammas will accumulate and say, hey, like there's no one else to play with, so we might as well like interact with each other, and they start forming tetramers. Beta thalassemia minor, we're going to have less beta chain, so that is certainly going to affect the A. Now, alpha, A2, and F are actually going to go up a little bit, those values, because remember, they're alpha, but they don't need beta, right? The A2 is going to need delta, and then the fetal is going to need gamma, and so those values are going to go up. So you can know it's low and low, or maybe just know the fact that both of those values go up, however you want to think about it. Nothing here. Really, this is only going to be for these two, so we can actually now ignore this entire section and this entire section. So beta thalassemia major, we really don't have any beta production or almost no beta production. So it's going to be like none or low for the A. A2 is trying to pick up the slack. It might go up a little bit. And the fetal hemoglobin might pick up the slack again too. Remember, there's no beta in the hemoglobin F. It's just the alpha. So you will have compensation with the HbF and HbA2 in the beta thalassemias as opposed to the alpha thalassemias. Remember that for the beta thalassemias major, this is Cooley's anemia, and you'll get these skeletal malformations in the face called, sometimes they call it a crew cut appearance, and it's, Cooley's anemia. I would be familiar with the physical presentation. Sickle cell is a hemoglobin defect where glutamate has been converted to a valine, and valine is nonpolar, and if one valine sticks onto another valine, they can hydrophobic interactions, they can start to accumulate. So what you get is these malformed hemoglobins that now have a hydrophobic component that can stick with other hydrophobic components, and they can aggregate together, forming these large clumps. When you have these large fibers of, like polymers of hemoglobin, it's not supposed to be a polymer, it distorts the shape of the erythrocyte, and it kind of looks like a sickle, hence the name. Obviously, changing the shape, the morphology of a blood cell is going to affect its ability to, move freely through the plasma and adequately deliver oxygen. People can have sickle cell disease where they are homozygote for the defective allele, or they can be heterozygous where they have sickle cell trait. Kind of like thalassemia trait is when it was kind of the intermediate phenotype. And so similarly, sickle cell trait is not as bad. So this picture is just showing that, the sickling is actually more severe in situations where, if somebody's exercising really hard, they're in a low oxygen environment in the tissues, that actually promotes sickling. So you can see that whether you have sickle cell anemia, homozygous, or sickle cell trait, heterozygous, where you are at a 100% oxygen, you don't even need to be 100% oxygen here, even at 70% oxygen, you're not going to have any sickled cells. But as we start moving lower in the oxygen saturation, we can see that this starts very quickly increasing the amount of sickled cells that we have. The sickle cell trait that requires a lower oxygen concentration get an equal amount of sickled cells as the sickle cell anemia. Nevertheless, both of these will have sickled cells. Oxygen makes it worse, and it's worse for the homozygous than the sickle cell trait. People are going to have occlusion of their blood vessels if you have these sickled erythrocytes. They really can plug up the smaller capillaries, making it more difficult for other erythrocytes to get through. And this can cause, extreme pain, this anemia. If you occlude the vessels, this can contribute to a stroke. splenomegaly, so defective red blood cells like we saw with some of the thalassemias. These need to be destroyed by the spleen, but if you have too many damaged, defective, sickled red blood cells, the spleen is trying to eat all of them and destroy them, and that is going to fill it up, and that's where the splenomegaly comes from. And as we mentioned, poor O2 delivery, you're going to have severe fatigue. Similar with thalassemias, or at least thalassemia beta, the treating with hydroxyurea can induce the fetal hemoglobin production. Somehow this inhibits the polymerization in the low oxygen environments. There's a couple different ways you can have sickle cell, but I would know, again, that it's mostly a glutamate being converted into a valine, and this hydrophobic interaction can cause polymerization. So if we look here now, considering sickle cell, all hemoglobin defects, both sickle cell and thalassemias, let's go ahead and look at a normal patient. We're mostly hemoglobin A. We have a teeny bit of A2, maybe like 2 or 3%, and maybe like 0.1% hemoglobin F. If we look at patient 1, we see that there's less hemoglobin A. There's more hemoglobin F and more hemoglobin A2. Now remember that A, F, and A2 all have the alpha chain, but only the hemoglobin A has the beta chain. So if hemoglobin F and A2 are becoming more common, it must not be an issue with the alpha, because they also have alpha. It must be an issue with the beta. And so it doesn't look, and especially if you look at patient 1 versus patient 2, it does, there's gradations of this. So I would guess that patient 1 is going to be a beta minor, and then patient 2 is going to be a beta major. If we look at patient 3, we see that there's a hemoglobin H. Remember that hemoglobin H is when you have 3 of the alpha alleles missing. So you only have 1. And in this case, you're going to have lots of beta chains left over, right? The stoichiometry, is a little bit screwed up. And so the beta chains will start to stick together. They form hemoglobin H. You'll get these target cells. And A should go down significantly. So this is H disease. And then patient 4 has hemoglobin S. This is just the sickled hemoglobin, probably where glutamate's converted to a valine, and so this is going to be the sickle cell disease. Parkinson's is a movement disorder caused by a destruction of the substantia nigra, which is in the basal ganglia of the brain. Recall that basal ganglia helps coordinate motor activities. So that's why this is a movement disorder. Destruction of dopaminergic neurons, and you're going to have this aggregation of the protein alpha-synuclein are going to be characteristics of this. The alpha-synucleins, as they aggregate, are going to form what's called a Lewy body. So alpha-synuclein helps regulate, dopamine is the neurotransmitter here in the presynaptic terminal. Its exocytosis into the synapse is regulated by alpha-synuclein, and when you have alpha-synuclein defective, you don't have that exocytosis working properly, and the vesicles are destabilized. Dopamine gets out, and it's causes lots of oxidative damage, which is one of the reasons that the neurons here might die in the substantia nigra. These alpha-synuclein proteins misfold, start to aggregate, and then it forms what's called a Lewy body in the neuron. And just things to look for, you get a tremor. So sometimes this is called a resting tremor, because if you're just sitting down, resting, that's when the tremor will happen. If you actually move to grab something, the tremor might get better. Sometimes it's called a pill rolling tremor. It looks as if they're moving their thumb and their fingers sort of together, as if they're rolling a pill in between their fingers. People become very rigid. Bradykinesia and akinesia, it's difficult for them to move and coordinate movements. And this is going to affect their posture and balance. So they have kind of this like shuffling gait where they're just moving very slowly, just barely moving one leg in front of the other. Well, if the issue is that the substantia nigra is damaged, and this is just something that produces dopamine to help regulate the movement. Well, then treating with L-DOPA, remember that phenylalanine becomes tyrosine and can eventually become DOPA, becomes dopamine. If you give somebody DOPA, this effectively can get into the brain. It can get through the blood-brain barrier, and it can help restore dopamine levels. So this is going to be the effective treatment here. There's lots of medications. This is the main one I would know for MFM. Alzheimer's. All right, so Alzheimer's is characterized by amyloid plaques and tau proteins. So you have something called APP, amyloid precursor protein. It's this transmembrane protein. And it has two different ways it can be cleaved. It can be cleaved via an alpha secretase, and this is the good, happy face way that it can be cleaved. Nothing's wrong with that. Or it can be cleaved alternatively through a beta secretase, where we're just, it's making a cut at a slightly different point. This is the bad way, because the beta secretase, I guess they're both subsequently cut by a gamma secretase, but again, this is fine. But the way that the beta cuts it, the secondary cut after this with the gamma secretase creates this protein derivative that can start, just like we saw with the alpha-synuclein, it can start aggregating and clumping together, and it forms these plaques. So because it's going through the beta secretase, we're going to call these beta plaques. So often when proteins aggregate together, that's just an amyloid plaque, and this is the beta amyloid plaque. You see this extracellular in the brain. So if you just think of all of the ways that, sometimes they like to either test your knowledge of medications that you've already known, or sort of like pitch a medication. So it might just be useful to think through this. A lot of different ways that the mechanism of action for drugs against Alzheimer's, there's actually not really any good ones on the market. You could have something that inhibits beta secretase. That would be great. Or you could have something that activates alpha secretase. Or maybe you have something that empowers the gamma secretase to cut the beta secretase derivative shown here in a slightly different way that is less likely to form aggregates. Or, beta secretase works as it does, gamma secretase works as it does, but somehow you get this thing and you somehow prevent the aggregation. Or you could take the plaque, and what if you could sort of program the immune system to destroy these plaques? These are all just theoretical ways which you could decrease the amount of beta amyloid plaques and maybe forestall Alzheimer's disease. All the different ways we could do it. Alpha-2 macroglobulin, this can help bring amyloid plaque into neurons where it can actually be more easily degraded. Remember, it's extracellular, but if it's intracellular, we could send it to a proteasome or something, maybe. And then the apo alleles. So there's ApoE is going to be an apolipoprotein, and that does a whole bunch of stuff with fat mobility and distribution throughout the body. But it weirdly has this side role where it helps regulate phagocytosis and glial cell activation. So phagocytosis would be good. We want, the microglia, sort of like the macrophages of the brain, to gobble up all of the beta plaques. And glial cells, they have something called reactive gliosis, where effectively they're creating like all of this scar tissue in the brain, and it's actually damaging the brain. So we want phagocytosis, we don't want glial cell activation. And you have two different alleles for two relevant different, there's actually many different alleles, but there's two ones that are important for the ApoE allele. Number 4, and this one is bad. So this is going to decrease phagocytosis and increase glial cell activation, which are all the worst things, right? So we're gonna have more plaques because we're not phagocytosing them, and glial cell activation just increases inflammation, and we have an accumulation of these senescent cells in the brain. I always thought like 4 as in forget, and then ApoE2. my girl, I asked her if she could think up like a clever mnemonic for this, and without missing a beat, it's so silly. She the North, like if you watch Game of Thrones, the North remembers, and she said it so matter-of-factly. The North is 2 words, and so 2 words and remembers. It's kind of a bad, that was cute, but it's kind of a bad way to remember it, but for whatever reason it stuck for me. So ApoE2, the North is 2 words and remember. So ApoE4 is bad, more Alzheimer's, ApoE2 is good, less likely to get Alzheimer's. Why is there a link with Down syndrome and Alzheimer's? Well, remember the amyloid precursor protein is the thing that can be cleaved via the beta secretase, gamma secretase, and then that can aggregate to form these extracellular plaques. Well, we got all these chromosomes. Which chromosome does amyloid precursor protein sit on? It sits on the 21st chromosome. And that is why they're going to have 50% more-ish APP, and so that makes them have more extracellular beta amyloid plaques, which means Alzheimer's. There's another protein, though, the tau protein. So, here's the soma, the cell body of our neuron, and here's these presynaptic terminals where the neurotransmitters sit in the vesicles. Well, how do we get them there? We have these long microtubule tracks, which can move things from the cell body where they're created to the end of the presynaptic terminal. So the microtubules are quite important in the axon. And what helps regulate and stabilize these microtubules is the tau protein. And if you over-phosphorylate a tau protein, for whatever reason, it causes it to let go of the microtubule, which means the microtubules can die. And also those proteins, then those tau proteins that are hyper-phosphorylated can start to stick together. They aggregate as well. So a lot of these brain diseases are just about protein aggregation. I would know that this is an intracellular aggregation as opposed to the beta amyloid plaque, which is an extracellular aggregation. The cerebral cortex, so like the outer part of the brain, the important part, I guess is all important, but it's going to cause it to atrophy and shrink. And the hippocampus, which is responsible for taking memories and sort of processing them and helping convert things you learn in a normal day into long-term memories, that is badly damaged, which is why memory is a particular noticeable clinical presentation for Alzheimer's. The ventricles, which contain cerebrospinal fluid, are going to enlarge a little bit in Alzheimer's. And just you can see that over time amyloid plaques, these come first, and then tau tangles, and then cognitive decline at the end. I don't actually think you need to know this order, but I found that interesting. Prions. So prions are really just these zombie proteins. So just a couple different terms. The main prion disease that we see in humans is the Creutzfeldt-Jakob disease. And this is where you might have we all have this normal protein in our brain. And actually it's right here. I would think like C for control. This is the nice healthy protein. I don't even know what it does. It's in our brain, it's fine. But if I ate a infected cow brain that had this misfolded protein, I think of it as a zombie protein because zombies bite other people and then turn them into zombies who bite others. There's kind of this positive feedback thing. So if I somehow eat a food and it gets a zombie protein from the cow into my brain, the zombie protein shown here is going to convert my healthy protein into an identical zombie protein. Well, now this zombie protein can go ahead and convert another normal one into a zombie protein, hence the positive feedback autocatalytic process. There's a couple different ways this can happen. Actually, everyone knows mad cow disease and hears about Kuru, but I think like 87% of Creutzfeldt-Jakob disease are just sporadic, meaning just dumb luck as you get older, just one of your proteins just gets misfolded one day, oops. But it's now a zombie protein and it turns, it kills you within like a year because of this positive feedback effect. So mostly it's sporadic. Now the famous cases are the mad cow disease, also known as variant Creutzfeldt-Jakob disease. That in like the 1990s and early 2000s, I think there was a bunch of infected cow. I think they were feeding cows food that had other cows in their brains until one of the cows had it, and they get transmitted everywhere, and then we eat the cows, etc. Kuru, I think in like New Guinea, there was some ritual where you would eat the brains of your dead ancestors or something. I'm sure I'm butchering that. I apologize. But if you're eating the brains of the people and they have an infection, and then you might get the infection. And so that's a very similar thing as well. So mad cow disease and Kuru are variant Creutzfeldt-Jakob. The sporadic is just where it just happens automatically. That's actually the main one. And then scrapie, if you hear about scrapie, and that's SC is actually for scrapie. This is because initially they found this in like sheep I think sheep and deer is this like contagious, version. Basically, the mad cow disease that we see in cows also happens in sheep and deer, and they call it scrapey because I think it affects the brain, but it made them really itchy for whatever reason. So you would see these sheep just they'd have giant bald spots because they were just rubbing themselves against trees and fences and stuff, and because they were so itchy, so they're scraping themselves against things, hence the name. And it causes these sponge-like lesions, these little holes in the brain. Michaelis Menten and inhibitors. It's definitely a high yield thing on the MCAT. And so if you know the MCAT stuff, then you're absolutely great on this. But it's really tricky. It confuses a lot of people, so I'm going to go into a little bit of depth on this. Here we're looking at a chart that just shows that the activity, the velocity of an enzyme, meaning like how many products it can crank out in a second or a minute, a certain period of time, is going to be dependent on the substrate concentration. Where you have more substrate, we're going to be able to generate more product per second. We're saying, okay, here's a certain substrate concentration here, and here's a certain velocity there. And if we increase the substrate concentration here, I'm going to higher velocity there. And what you should see is that this kind of plateaus. Okay, why does this plateau? And I would just know this general thing. Enzyme plus substrate makes enzyme substrate complex. So it has to sit together. I always thought of it as I'm gonna bore you to death with this analogy, but it's the car salesperson and it's the person trying to buy the car. They're just wandering around the used car lot. Here is them actually interacting, and the enzyme and the substrate are together, the salesperson and the person trying to buy the car together, and we're trying to close a deal here. And this takes time, this takes effort. And then eventually, maybe, hopefully, the car salesperson can go back to trying to find another customer, and we now have a product. So a car has been sold. What we'll notice, though, again, is that this kind of plateaus and why. If we think of these are our employees, and these are the customers right here, if you have 3 employees, and how many do we have here? A low substrate concentration, 1, 2, 3, 4, 5. A lot of customers don't want to be bothered, and a lot of employees are lazy. So it might just be that some employees aren't trying to sell cars. So our velocity is going to be kind of on the lower end here. We're not going to sell that many cars. And it's because a lot of our employees just aren't interacting with anybody. But what happens if I have way more customers on the used car lot? Well, there's a higher likelihood that the employees, the enzymes, are going to be engaged with the customer, trying to make a sale and close the deal. And so, now we're going to have a little bit more product being made, cars being sold. Okay, what if we just have a ridiculous amount more customers than we've ever seen on a used car lot? In fact, you know what? We have too many customers. We have so many customers that, look, every single one of our employees is engaged with the customer. We couldn't do better, right? This is the best we could possibly do. We might say that we're saturated here, right? If you double the amount of customers, well, you're not going to sell more cars, because to sell a car, you need to go through the employee, right? It takes time to make the sale and do the paperwork, etc. So you would probably need more employees, or faster, better employees, which we'll get to if you want to increase the velocity. But we call this the Vmax, the most cars you could possibly sell in an hour, the most protein you could make in a second or a minute or whatever. I would know the Michaelis Menten curve. So this is, we could go into more detail on this, but let's not. Velocity, this is how many proteins you make, how much product you're making per second. This is equal to the maximum product you could possibly make given the amount of enzymes you have, times the substrate concentration, whatever it is, over Km. And the Michaelis Menten constant, we'll talk more about this, but I would just know, if this is my Vmax, this is 1/2 of Vmax, and it corresponds with the 1/2. So the 1/2, it is the concentration of substrate. In other words, the amount of customers you need on your car lot to reach half of the maximum amount of sales you could ever hope to make. So it's the substrate needed to get to 1/2 Vmax. All right, bunch of different ways to think about Km. I would think of it as the infinity that an enzyme has for a substrate. If you have a really low Km, then that means that the protein is really, really magnetic, high gravitational attraction for the substrate. High Km means it's a lower affinity. What, like we just said, maybe the second one, it's the concentration of substrate. We're at half of the maximum velocity. And then this third one, this one's really mostly just useful for when you're thinking of uncompetitive inhibitors. So we'll come back to this, but this is it is quite literally the stability of the enzyme substrate complex. I know that one's kind of weird, so put that in the back of your head. We'll come back to that. And for our dumb metaphor, we're going to think about is how enthusiastic and hardworking is our salesperson. Like how eager are they to find a customer and try to make a sale, right? Now, no, that's way different than saying they're actually good at making a sale, right? They might be eager, but just terrible salesperson, right? So let's disentangle those two concepts. Vmax. So we said that this is the maximum amount of product that our proteins can make in a given second or a given time period, and it's dependent on K2 and the total amount of enzymes, total amount of workers you have present. So Vmax is equal to K2 times the total enzyme concentration. And then if we think of our car example, let's say that the most cars we could sell in an hour is 10. Well, that's going to be dependent on how many employees do we have, how many enzymes do we have, the total amount. And K2 is like how good of a salesperson are they, right? And how quickly can they do the paperwork and whatnot. So K2 is like how competent they are. And then, and then that kind of makes sense. Like if they're really good at doing paperwork and making a sale and you have tons of employees, then you'll have a really large Vmax. And like we just said, we can think of it as the amount of cars that we could sell, the maximum amount, right? V is just the literal amount depending on how many customers, how much substrate we have at any given time. All right, you get it. We're talking about cars. All right, so a low Km is a can-do attitude. So it's a high affinity for a customer. It's a salesperson who's going to hustle their ass off. So even if there's few customers on the car lot, it doesn't really matter. They're meeting with them and they're trying to make a sale. So someone wants help and they're like, yes, boom, I'm here, I'm eager, let's do it. High Km, the salesperson can't be bothered. They hate their job, they hate their life, they don't want to help. So someone says, can I get help? It's just one person. And it's like, it's just one person. I'm on break. I don't want to interact with you. Like, I have better things to do. However, if there's like 1 million people all saying, hey, I need help, there's just, there's 30 people and not that many salespeople. The salespeople are like, ugh, fine, there's just so much demand, even though I don't want to. Now I'll help you, right? Because like you're in my face. I tried hiding in the coffee room. I tried hiding in the bathroom, but the customers are relentless. They found me and they're forcing me to help them try to buy a car. That is different than K2, also known as K-cat. This is about charisma, right? It's nothing to do with laziness or hustle, This is once you have a customer in front of you. In other words, once you're in the enzyme substrate complex, how good are you at generating a product, right? In other words, how quickly and how able are you to close the deal? So a low K2 employee just, lacks a certain, finesse where, someone says, hey, is there any wiggle room on the price? And then they say something stupid, right? So they're not going to close this deal, or it's going to take them a long time to close this deal, right? On the other hand, high K2, very charismatic. They're going to say something smooth that is going to make the the deal go more quickly. I think you get what I'm saying. All right, so again, I'm going to really hammer this in. Km and K2, let's not over anthropomorphize it, right? There might be a correlation between a hardworking employee and a charismatic one, but we're pretending like they're completely different things, right? You could have a high Km, high K2, low Km, low K2, high Km, low K2, or low Km, high K2, right? You could have any four of those combinations. And the best employee is going to have a low Km. They're good hustle. They really want to engage with the customer. And they also have a high K2. They have a high charisma. And again, Vmax is the number of employees we have and how charismatic they are. So it does not rely on Km at all, as we'll see. So let's go ahead and look and see, if we look at different, K2s and Kms, how does this affect, like, how many cars we're going to sell? All right, this is a reference dealership. And notice that, so in every one of these we have 10, 10, 10, 10, 10. We always have 10 salespeople. I'm not changing the number of salespeople in these examples. So that's not really a factor. Really what's changing is just how many customers we have on our car lot. We have 2 and 20 and 50 and 100 and 500. And so why have I highlighted this yellow here? The idea is when you have 500 customers versus 100 customers, you're only selling 10 cars. So you know what? Whether we're here or whether here, this is our VMAX. This is the most cars that we're going to sell, right? If you have 10,000 customers, you're not going to sell more cars because you only have a certain number of employees, and they can only interact with one customer at a time, right? What's our VMAX? It's 10. What's our KM? Well, remember, KM is the value. So if this is 10, well then 5 cars sold in an hour is going to be half of our VMAX, and that's going to correlate with that number there. And we actually know this number, right? 5 cars sold correlates with 20 customers. So these are the two values. VMAX is 10, KM is 20. All right, so now we have charisma is the same. K2 is the same, but they're really lazy employees. They do not want to be bothered. They just don't. They're hiding from customers. They're just trying to get through the day with doing as little work as possible. Now, once they're interacting with the customer, they may or may not be good, right? But we're just saying they're lazy. So again, there we go, fun. 10 cars sold, that is our VMAX. That's the same, right? But let's go ahead and see where is the 5 cars sold. It's right here with 50 customers. So now what's our KM? It's 50. So we have kind of a lower affinity, a lower willingness to bind with customers, to bind the enzyme, to bind with the substrate. And then the opposite of this is that, again, fun. We're going to have 10 cars sold, but we've got the eager, like, the last one was Squidward and this one is SpongeBob, is they're dying to meet a customer and try to close the sale. They live for this. And we're looking for 5 cars sold, which, would be in between this and this. And so, we're going to say that it's like 10-ish. So notice in those 3 values, we had the KM was 20 for the reference, and the KM was 50 when it was the lazy employee, and the KM is like 10-ish when it's the very enthusiastic employee, right? So again, low KM means high affinity. You're very quickly going to ramp up and get to your highest possible, sales point, your highest VMAX, right? Doesn't mean your VMAX will be high. It just means you're going to get there really quickly. I mean, imagine a car that can like accelerate to, maybe the max speed is 100 meters or 100 miles per hour, right? You can accelerate to that really quickly. That is going to be a low KM. If you accelerate to that very slowly, that's going to be a high KM, right? It doesn't mean that the max speed is 150 or 50, it's 100 no matter what. Oh gosh, what am I talking about here? Okay, so yeah, what if we had more employees, or what if we had better employees? I think in this example, we're mostly going to be talking about better employees. But notice that it's really the same if you're talking about K2 or E total. Increasing either one of these would increase the amount of cars that you're going to be selling, your VMAX. But we're mostly going to be talking about better employees, more charismatic. So we're going to be looking at a bad salesperson versus a good salesperson. Again, we are not talking about how willing they are to meet a customer, just how good are they at closing a deal and how quickly can they close a deal. So again, fun. We have our 10 cars sold. And so what is our VMAX? It is 10. What is our KM? Well, 5 is going to be half of that, and that's 20. Again, this is the same as it was before, 10 and 20. So here you can think of this as less employees, or maybe let's just think less charismatic. So equal number of employees, but all of them are less charismatic. So they're just going to waffle, and they're just going to blow the deal when it comes their way. And so now we're seeing that the most cars you could sell is 6. Our VMAX is 6. And that's because we have these bad salespeople, or it takes a long time to close the deal. But notice they're just as willing to meet a customer as earlier, right? They're not more or less willing, it's the exact same. And so what we see is our half of 6 is 3. So we actually have the exact same KM as before. It's just our VMAX has gone down. And what if the opposite happens? So now we have more employees, or just more charismatic employees. Again, we have 20 cars sold, and half of that is going to be 10 cars. and that's going to be associated with 20 customers. Again, the KM has not changed, right? It's just the Vmax that changed. And so Vmax would be 20 and KM is 20. So as we mentioned, a good employee is a high K2 and a low KM. So I think this is just the best way to think about K2, Vmax, KM. I know it's silly and I'm certainly beating a dead horse, but I just think it's a really useful concept and it certainly could come up on your NBME, especially with how it relates to inhibitors. So like if you have a good understanding of KM and K2 and whatnot, it makes it a little bit easier to understand inhibitors. So competitive inhibitors, we have our active site right here, normally where our substrate goes, but if the inhibitor looks like the substrate, it could easily fit into that spot. So it's going to mimic the substrate, and it binds to the unbound enzyme, right? This is the employee before they've met with the customer and tried to make the sale, right? They're just walking around on their own. That is the only time that this imposter substrate can get in. It can't get in for the enzyme substrate complex. Non-competitive, it doesn't matter. The inhibitor can bind somewhere else other than the active site, an allosteric site. It binds anywhere else. And it doesn't matter if the substrate's bound or not bound. That has no bearing. So it can bind to the unbound enzyme or the enzyme substrate complex, so when it is bound. Now, uncompetitive here, when it cannot bind in this situation, which seems weird, right? Because you're like, wait a second, it's an allosteric site. Why would it not be able to go there? Well, when the substrate gets into the active site, a lot of times this induces a conformational change for the protein. And if you actually look very carefully, this does not quite match this little curve here. But here what we've done, instead of this right there, we now have a little curve here. Why did that curve show up? Because this binding induced a conformational change, which made this curve happen. So it is only in the correct conformational shape for the inhibitor to bind after the substrate is bound. So in other words, long story short, the uncompetitive inhibitor can only bind to the enzyme substrate complex because the shape of the protein is a little bit different in the enzyme versus the enzyme substrate. Competitive inhibitor, if we're just, I'm gonna keep going with this silly metaphor, but you can imagine that like a rival car dealership sends over like actors. And okay, why would that help like hurt our business? Well, if I have a car lot and maybe there's like 8 people walking around who I want to sell cars to, if like 3 of them are actors, imposters, and their only job is to waste my time, they talk with my employees for like 3 hours. They pretend like they're going to go buy a car, but they have no intention of buying a car. That's going to waste my employees' time and it's going to affect our bottom line, right? We're going to have our velocity is going to be lower. On average, we're going to be selling less cars per day. But if you think about that though, the Vmax should stay the same because we said like an average day, right? When I have like 8 customers and 3 of them are imposters. But what if I had 10,000 customers on my used car lot and 3 of them are imposter actors? Well, I only have like 14 employees. I have 10,000 people in my car lot, 3 of them being competitive inhibitors, imposters. It's just the likelihood of everybody's trying to get the attention of the employee. It's just a really, really low likelihood that they even interact with the imposters. So if you have a high enough substrate concentration, enough people wanting to buy cars, you probably just won't even interact with the competitive inhibitors. And so they will have no effect. So you can kind of drown it out. But the idea though is it does take more people to get to that Vmax. And if it takes more people to get to that Vmax when you have these imposters, then it also takes more people to get to your KM, which means your KM has increased. So KM has increased, but Vmax is the same. And these are all just pictures of, here's folic acid, but you can notice methotrexate, leucovorin, things like aminopterin all have very similar shapes. And so these are classic competitive inhibitors. So you should just be able to eyeball 2 things and be like, they kind of look similar. It's probably a competitive inhibitor. Non-competitive, so for lack of imagination, this is like you just have a murderer, right? Okay, the rival car dealership just sends over an assassin to murder your employees. It doesn't matter if the employee is interacting with the customer or not interacting with the customer, right? Whether in the unbound E or bound enzyme substrate ES complex, they're just going to murder people. And you know what? If you're murdering my employees, that is less employees, my Vmax is going to go down. All right, this one's weird, but imagine that they're assassinating the employees, but the other employees aren't finding out, right? It's probably bad for morale if you know people are getting murdered, but we do not know people are getting murdered. And so if we're losing our employees here, but the other ones don't know about it, the KM is going to be the exact same, right? The other employees are just going to go to work, do the exact same job. The KM does not change. That is kind of weird, but the KM stays the same because I'm hurting one employee, but I'm not affecting the other employee. The unaffected employee's KM is the same. And uncompetitive. So here, our last example is imagine that we have the salesperson here and the employee here. They're interacting, trying to close a deal, trying to convert a substrate into product. But now the uncompetitive inhibitor can bind. And this is a different picture than earlier, but it's the same concept. The substrate binds, changes the shape of the enzyme, and now, and only now, can the uncompetitive inhibitor come in and bind. It locks the substrate and the enzyme together. It ties them together with a rope, effectively, so that they cannot dissociate. And you know what? It's kind of difficult to close a deal if you're tied together with the customer. That's going to make it a little bit weird. So your Vmax is going to go down. You can also just think of it as like you're taking an employee out of commission. They can't do their job. So if you look, Vmax is equal to K2 times the total concentration of enzyme. We have less enzymes, and therefore we are going to have a lower Vmax. What's weird though, and remember we had so many different definitions of Km, is that your Km actually goes up, right? Because Km really represents the stability of the enzyme substrate complex. And in other words, how easy is it for them to dissociate? And if they're tied together with rope, it's very difficult for them to dissociate. So a high Km actually means that it's very stable because they can't dissociate because the uncompetitive inhibitor is locking them or tying them into place. So the Michaelis Menten formula, if we do a bit of algebra voodoo, we flip everything, etc. This can be my y. This can be my slope m. This is kind of hard to see, but we can think of this as times 1 over s. This is 1 over s. 1 over s is our x, and then 1 over Vmax is our y-intercept b. So this is the Lineweaver-Burke equation, and it's just a nice way to quantify things like a little bit more objectively. So if we look at this is our y. So this is the y-axis. It's 1 over v. Km over Vmax is our slope. So that is our slope. Rise over run, change in y over change in x. And then 1 over s is our x. And so that is our x-intercept. Our x-intercept is 1 over s. And then 1 over Vmax is where our line crosses the y-axis, the y-intercept. Now, this one is important to know too. It's kind of weird. The x-intercept is actually negative 1 over Km. Why is that? Well, I don't feel like writing it out, but if we're saying that, okay, the x-intercept is where y is 0, if I made this 0 and I set it to Km over Vmax times, let's just call this x plus 1 over Vmax. So again, we just made 1 over v 0 because that's where the x-intercept is. And instead of writing 1 over s, I just wrote x because that's what it is. If you solve for x, it will be equal to negative 1 over Km. Just take my word for it. So let's combine all of this. So competitive inhibitor only goes through the enzyme. Km is going to go up because you need more people on your car lot to get the half of the maximum sales, right? Because you got to deal with those imposters wasting people's times. But the Vmax is the same because you could drown out, flood out the imposters. If you just have 10,000 million, billion, trillion customers on your lot, it's just really unlikely that the employee is just statistically, they're unlikely to interact with the imposter. The assassin does not discriminate. Enzyme substrate. It's going to go for both of them equally. Km is going to be the same because like the employees that aren't being murdered, they're going to have the exact same. Willingness to engage with employees, but your Vmax is going down because you have less employees because they're being murdered. Here, we're only binding together the employee and the substrate together with rope. We're not going for unbound enzymes. And Km, this should actually say, oh gosh. So earlier when I said, if I said it goes up, I misspoke. Km is going to go down, but down nevertheless means that you're going to have Km should go down. That's a typo. Km goes down. Vmax also goes down. They both go down. But nevertheless, it still means that you're more stable when we say that the Km goes down. So sorry about that. So here the Km is going to go down and the Vmax is going to go down as well, because you know what, you're also taking, it's kind of like murdering an employee if you tie them up. We have one less employee to do their job. So let's look at all of these. Competitive inhibitor, the Km is going to go up, the Vmax is going to go down. So if we imagine like this is -1/1, so this is -1 right here. If the Km is going to go up, instead of -1/1, I'm going to have something like, you know, now it's going to be like -1/2, a higher number. -1/2 would be, I don't know, like right there. And the Vmax is going to go right there. And so we're going to get something like that. So I was close, but it's going to be like right there. Yeah. And the slope would go up as well, right? Because it's Km over Vmax. Km goes up and the Vmax stays the same. And so that would be higher slope. Non-competitive inhibitor, the Km stays the same. Okay, so that's easy. The x-intercept is the same. But the Vmax goes down. Okay, well, let's imagine that this is 1/1. Great. Well, if that's 1/1, the Vmax goes down. What's 1/0.5? It's something like 2. So it would be like up there. So we need something that goes there to there, and we get something, dingus, it's close. It goes up there, right? And again, we should have a higher slope, because the slope is Km over Vmax. The denominator is getting smaller. That's a higher slope. All right. Uncompetitive inhibitor, tying them together, Km goes down. So again, I misspoke earlier. Km goes down. That means it's more stable. They're locked together. So if this is -1/1, that's -1. Now the Km goes down. This is going to be -1 over, like, I don't know, 0.5 is going to be -2. It's going to be like over there. And the Vmax is going to go down. If this was 1/1, but now it's like 1/0.5, that's going to go there. And you know what? Let's think about the slope. The slope is Km over Vmax. If they both go down, and it's actually important to know that they both go down an equal amount, my slope should stay the same. And actually, I was close. I'm never going to get this. But nevertheless, so the easiest way to see this is that the slope stays the same. And but all of these, like the slope, in my opinion, is an afterthought. If you know where the x-intercept and the y-intercept should be, you should be able to figure out automatically what the slope is just by connecting the dots. Okay, pivoting over to sugars now. So sugars, really just all carbohydrates. You think about the name, it's carbon and a water, and however many of them you have, that's really just the formula for a carbohydrate. So CH2 is, going to be the first one, and then you have C2H4O2, and then we can go all the way to the more familiar C6H12O6, which is things like glucose, fructose, galactose. Of all of these sugars, carbohydrates, when they're in the linear form, if you have a carbonyl that is really just an aldehyde, that is an aldose, the name is fitting. If that carbonyl is part of a ketone, then we call it a ketose. A lot of times these sugars, though, are not going to stay in the linear form, in the Fischer projection. They're going to sort of link around and become cyclical. We're going to call this a Hawthorne projection now when we look at it from a cyclical perspective. If it's a 6-membered ring with an oxygen as an ether, that's a pyranose. And if it's a 5-membered ring, that is a furanose. I would just maybe know that fructose is often going to be a ketose and a furanose, and most other things are aldoses and pyranoses. So like we just said, that you can depict it here in the linear chain, but you can also show that, here it's technically still in the linear chain, but we've just bent it so it's a little bit easier on the eyes. It's, they're never really in three-dimensional space in reality gonna look like we see right here. It's going to exist something a little bit more like this right here. And what's going to happen is if we count down, assuming we're starting with an aldose, here we have D-glucose. And by the way, you can know it's the D form. So most sugars will be D, and most amino acids will be L. And really these are just different enantiomers, which derive their name, D or L, versus whether dextrotary, lev rotary, whether they rotate light one way or the other way. I don't think you really need to know that too much, but maybe be able to notice that this is a D sugar, because when we look at the last chiral carbon, so carbon, chiral, 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 not chiral. So the last chiral carbon, the OH is going to the right in the Fischer projection. I always thought of in like Spanish, D derecha means to the right. And if that OH goes to the left, well, then L for left would be the L-glucose version. Notice that when you do go to D to L, though, it's not like the name comes from the last chiral carbon, but every other chiral carbon does have to flip. Anyways, back to the intramolecular reaction. So the last chiral carbon, this one right here, is going to react and come in right there and nucleophilically attack this carbonyl and open it up. It's much clearer here in this picture. And when it does attack, so this bond is going to flip over here, and you could have the OH going kind of like, think of it as above the ring or below the ring. Really, it's, if you think of chairs in organic chemistry, we have equatorial and axial. You can get the axial version, which is going to be the alpha glucose, or the equatorial one, which is the beta. So the beta is the OH is going to go up, and here the axial, the OH is going to go down. So as we said, sugars are D, amino acids are L, and we could get really in the weeds with this, but I would just know that the L and the D are enantiomers of one another. They are non-superimposable mirror images, and they're going to rotate light in different directions. And so, here we can see that the D, OH goes to the right. Here, L, the OH, the last chiral carbon, is going to go to the left. And you can see here, though, that it's not just, it gets its name from the last chiral carbon, but all of the other chiral carbons are flipping as well. And you can see that in the picture here. This is going to be like the sort of the normal state of affairs, the D version. This one looks a little bit wonky. You don't typically see the alpha glucoses, but notice that everything does flip in the Hawthorne projection as well as the Fisher projection. So if I have, let's say I'm looking at two molecules, and they're pretty much, they are identical in terms of their structure, but you're trying to figure out the stereochemistry. And one of the molecules is RRR, and the other molecule is RRR. You know what? If they're probably the exact same structure, right? So these are identical molecules, like two methane molecules. They're identical. So that would be an identical molecule. Okay. What if that same molecule now we flip all of the chiral centers. So now it's SSS. We've now created a perfect mirror. So this is an enantiomer. It's the mirror image version. So you could have two molecules. Are they the same molecule? Assuming that we're stereoisomers. Are they the same molecule? Are they complete mirror images? They're enantiomers. What if they're anything else? What if it's RRR is the first one, and then the next one is SS. So only a mirror image for the second part and the third part. Well, this isn't not the same molecule, and it's not a mirror image. So it just goes into this leftover like dump category called diastereomer. Diastereomer just means anything that's not the same molecule or not a mirror image. So that is what a diastereomer is. Diastereomer is kind of a broad, all-encompassing category. And what's an epimer? An epimer is a diastereomer where only one of the chiral center flips. So if I had RRR, and then I had like RRS, only one of them flips. That is an epimer. You also could have had RRR and RSR, right? That would also be an epimer. So we're seeing this glucose and galactose are the exact same. This is to the right, that's to the right, that's to the left, that's to the left, that's to the right, that's to the right. But this one right here is right, and this one here is left. That is a diastereomer. And even more specifically now, it is an epimer, because only one of them has flipped. An anomer is an epimer where only the anomeric carbon, anomeric carbon again is going to be the one that holds the C double bond O in the linear form. It is the electrophile for the OH attacking right here. So an anomer is when we already talked about this. If that goes down, it's alpha, and if it goes up, it's beta. These are anomers. Everything is the same except they're flipped at this anomeric carbon. So this is a diastereomer, check. This is an epimer, check. This is also an anomer. It's all three. And the beta one is more stable. So it's actually going to equilibrate back and forth. Maybe I have a better picture of that. Yeah. So if it's in the alpha, it constantly is going back between the closed form and the linear form, closed form, linear form. But it'll go from the closed form to the linear form to this form and back again. And it's constantly cycling between these three things. It probably spends most of its time in the beta ring structure. That is the most stable. If you think of your organic chem and your Newman projections, so this is equatorial, the beta, and the OH is the axial. It's 120 degrees away from the ring in the OH version. It's only 60 degrees, so there's more steric interaction here. You don't need to know that, but if anybody's like an orgo nerd, that's like a fun connection to make. And so yeah, what we often see in the drawings are not representative of three-dimensional reality. So it's hard to get just from this picture going up, going down, the idea of 120 degrees versus 60 degrees, but that is why it's more stable. A glycosidic bond, just a link between two sugars. Anytime we're linking up two monomers, we're usually doing a dehydration reaction. We're taking out an OH and an H, well, that's water. Now we've linked these together. This would be an alpha 1,4 bond. How do I know that? Because this is the anomeric carbon 1. This is the carbon that's attached to, not anomeric. It's just the first carbon is always going to be anomeric. So this is just some random carbon. So anomeric, just some carbon number 4. And where is the O going for carbon number 1? It's going down. We know that up is beta, down is alpha. So that's alpha 1,4 glycosidic bond here. And again, this is alpha 1,4. And if you had two glucoses together, this would make a maltose. Don't memorize that, but that's just what it would be. And you could have so effectively a cellulose structure if you go beta 1,4. It goes up and then over to the 4. All right, you get too much sugar, too much glucose in your blood is really just diabetes, right? High blood sugar is bad for you, and there's a bunch of different reasons. It can, convert into something called sorbitol, which absorbs fluid, which damages the eyes and Schwann cells and pericytes, but it can also cross-link proteins in the blood. And one of the most abundant proteins in the blood is the albumin made by the liver. So glucose can bind to, you got too much glucose, you got these proteins. Eventually, if the number is too high of glucose, they'll bind. This it's called a Schiff base. It's really just an imine, if you remember orgo. That, though, can then react, become a fructosamine, which is also known as an amidori product. And these can oxidize and become advanced glycation end products, also known as AGE. And these effectively, this is just proteins with these sugars on them, and they start cross-linking. And if you have too many proteins cross-linking, they might deposit into the the vascular tunneling, right, into the arteries, into the veins. This can lead to hyaline arteriosclerosis. It can lead to atherosclerosis, right? If we're pushing too many proteins into the blood vessels, they're now just, I'm waving my arms around, you can't really see, but you have these proteins which are pushed a little bit into the blood vessels, but they're still hanging out into the lumen, and they might catch on to these LDL lipoprotein particles containing fats, and then the fat will basically deposit into the blood vessel, and that leads to atherosclerosis. What you probably need to know for right now is it's bad. You don't want too much blood in your sugar. And then the hemoglobin A1C is actually a useful way. It's a proxy of seeing exactly what your blood sugar is. Because if I get a blood sugar test, let's just say I had, a crazy day, like, 5 croissants and a bunch of candy bars, and I test my blood sugar the next morning, it might be really high, and I think I have diabetes. When in reality, I just, it's not a good thing. I'm certainly not on the right path, but I just had too much sugar. But all the other days I was good. Normally my blood sugar is fine. Well, the hemoglobin A1C is good because it takes it looks at like the long haul. It's a few months snapshot because albumin is going to last in the blood for a couple of months before it's recycled. So a high hemoglobin A1C is maybe a better diagnostic marker, a better proxy for whether or not you have diabetes. So above 6.4, 6.5 is going to be in the bad territory. But on a more minute-to-minute thing, you can do blood glucose monitoring. You typically, to avoid I took my blood sugar after I had a candy bar effect, you're going to want to do a fasting blood glucose. So you're going to, not eat for, 8 to 12 hours, and in the morning you'll take one or get your blood drawn. Normal would be 80 to 100, but above 125 is going to be in the diabetic range. And this is just how this works. I don't really think that's that high yield. So a bunch of different mechanisms how this works. Too much proteins getting, the advanced glycation end products cross-linking and causing mischief in the blood vessels. You can get, conversion of, you can get generation of sorbitol, which is going to accumulate water and can cause the eyes to swell and damage the nerves and whatnot. So mainly diabetic retinopathy. It can lead to blindness, nephropathy. It can damage the glomerulus and affect the kidneys. And then neuropathy, which is like tingling in the fingers and toes and extremities. And often people will feel this in the mornings. And they call hypertension and diabetes are the silent killers because people can be asymptomatic for years and years and years, and then suddenly their vision starts to go because the blood sugar has been slowly damaging the tissue there. All right, glycogen. So the glucose is going to be more immediately accessible, but we want a storage form. So we can store glycogen in the liver and in the skeletal muscle. Remember that the liver is altruistic. So if you haven't eaten for days, you're in the middle of the forest, your liver is going to break down glycogen and push glucose into the blood altruistically to feed the rest of your body. Skeletal muscle, though, they're the assholes, the jerks, they're selfish, they're hoarding the glycogen for themselves. Kind of makes sense though, because if you want to run from a bear, that probably is going to take priority over like my tummy having a little bit of extra sugar to digest a picnic or something. So it's going to be maybe mostly alpha 1,4 bonds. So here is an anomeric carbon that's going down, so it's alpha, and this is carbon 4, so alpha 1,4. That's mostly what we're going to see, but we'll see a little bit of alpha 1,6. So this is going down on the anomeric, so that's alpha, but it's now going 1, 2, 3, 4, 5, 6. It's an alpha 1,6 link. So maybe 1 in 10 of these will be alpha 1,6, and that's really just to, increase the surface area, so I can fit more sugar into, increase the density of the stored carbohydrates. All of these are non-reducing ends. What is a reducing end? A reducing end is right over here, right there. And what we're going to do is look for the hemiacetal. So hemiacetal is where we have we have a central carbon, we have an OR, we have an H, we have an OH, we have an R. That is a hemiacetal, and that is what we look for. That is going to, so here we have exactly this, right? That's the OR. That's the R, that's the OH, and then that's the H, right? That is, it looks ugly, but it's the same thing. It's a hemiacetal. That is what a reducing end is going to be. Boom, right there. So back to orgo, which again, I hope I'm not freaking everybody out. I'm probably going a little bit more in depth than you need to go, but it's good if you know it. If you don't know it, it's not the end of the world. An aldehyde could react and become a hemiacetal, but this reaction is reversible, right? The hemiacetal can also go back to become an aldehyde. So let's imagine you have a hemiacetal. It goes back to become an aldehyde. And what can we do with an aldehyde? We can oxidize it. You oxidize it, it becomes a carboxylic acid. Why is that relevant? Because, well, if we look at glucose again, we have a little hydrogen here. And so this is my central carbon of the hemiacetal. Well, that can react, become an aldehyde. And really, it's going to do this by going from the cyclical version to the linear version. Kaboom, right there. So this reaction, which we showed in the earlier picture, now this is an aldehyde. We can oxidize this aldehyde. Glucose, or really any reducing sugar, yes, it's normally in the cyclical version, but it can quickly equilibrate and go to the linear version where it's an aldehyde, and then it can be oxidized. And that oxidized is going to be important, one, for a lot of enzymatic reactions, and it makes the molecule more accessible to break off, and et cetera. But I would say especially for testing of blood sugar and whatnot. So if we have a reducing sugar, it can become an aldehyde, and then the aldehyde can be oxidized. Well, and if it's becoming oxidized, it's reducing something else. We're reducing rocks. We're reducing some metal, whether it's copper or silver or something, and these will be things like Benedict's test, Fahling's test, Tollin's test. And this is in the reduction of this metal gives us a bright color. And the observance of that color is really just, if you have more of the bright color show up, it means you had more reduction of the metal, which means you had more oxidation of the sugar, which means you have more sugar. So the more color we get, the more sugar we must have had. And this is the underlying concept for a lot of these blood sugar tests. Galactose. So galactose disorder. So lactose, is really just a disaccharide, which is made up of glucose and galactose. And ideally, we have lactase to break down this kind of like milk dairy product into these two, and then they can go through these respective enzymatic pathways. Glucose, of course, goes to glycolysis. We'll get into that, but galactose can either go this way or it can go this way. All right. A lot of people don't produce the enzyme lactase, or you produce it as a kid and you lose it as you become an adult. This is something we evolved in the last 10,000 years as we became like, pastoralists with sheeps and goats and cows that could produce milk, and people who could digest milk for longer, right? Maintain this ability to transcribe and translate lactase from childhood into adulthood. This became selected and preferred in certain civilizations and areas. So some people don't have this though. And what happens if we don't have this? Well, if I'm not digesting the lactose, you know who is? The bacteria in your stomach. So the lactose is going to go to your GI system, small intestine, and we have a lot of bacteria here. Sorry, it's normally broken down in the small intestine. Maybe, yeah, in the small intestine. But then we're going to have a lot of bacteria in the large intestine. They're going to break it down. They produce a lot of gas, and this gas is going to be responsible for a lot of the symptoms: flatulence, abdominal pain, etc., belching, and no good. Galactokinase deficiency. So this is going to cause eye problems. So if we don't have this, right? So I'm still breaking down in my small intestine the lactose into glucose and galactose, but the galactose now cannot go to the left pathway. So it all is going to go into this right pathway. It makes galactitol. So I mentioned sorbitol earlier. You don't really have to know what that is yet. We'll talk about it more soon in upcoming lectures. But galactitol is going to be a very strong osmotic agent. It's going to pull in tons of water. And if you pull in too much water, you might cause a cell to swell and you might damage it. It's especially bad for the eyes. It can cause glaucoma and too much pressure build up in the eyes. So it causes eye problems. And there you go. You get this kind of cloudy, cataract and glaucoma and lots of other problems. For whatever reason, there's a large amount of aldose reductase in ocular tissue. So we're going to have a large buildup there, which means more fluid and more damage. So that's galactokinase deficiency. But what if we have classic galactosemia? In my head, I would have thought that if we're breaking this one down, okay, we're getting further through the pathway, so this is going to be better. Turns out this is actually worse. And I remember so this is also going to cause eye problems. We're not getting to the end product, so we're still going to get more of this. But I would say it's actually worse. It's going to cause developmental issues in kids. Why? Because we still have this galactokinase working. In kinases are going to take ATP and take one of the phosphates from it and stick it onto the galactose, but for nothing. This galactose 1-phosphate is not doing anything, so I'm just wasting ATP, slapping phosphates on galactoses that I will never get back. And so this is going to deplete a lot of the energy, the ATP currency of the cell from a young child. And additionally, the galactose can stay outside of a cell, but once it gets into the cell and you put a phosphate on it, you trap it into the cell. So not only are you wasting ATP, we're trapping it into the cells where we can't really get rid of it. You might be able to have other mechanisms to get rid of it if it's in the plasma, but here it gets into the cell, it can't get out, and you're wasting ATP. That's why this is the worst phenotype if you're allowed to do this first phosphorylation step with galactokinase. And I just thought of this clip and Silicon Valley, if you're doing a startup company, it's better to fail quickly and then start another company than to invest a lot in a company and have it fail a couple of years from now after you've wasted a lot of time and money. And I think of the same thing here. So this is what glycogen looks like. It has glycogen in this protein in the center. And again, we show that we have these alpha-1,4 chains going this way, but an occasional alpha-1,6 followed by alpha-1,4, alpha-1,6. And so this allows us a large density. You can squeeze in, right? Imagine if I had a central protein and it could only go straight. This is a lot of volume here that I'm not exploring. But if I can branch off, then I can get a little bit more of that. So we said alpha-1,6, we said alpha-1,4. That's all good. The reducing sugar is going to be somewhere over here in the end. Starch, when we're eating bread, if you have amylose, it's going to be all alpha-1,4. If you have amylopectin, it's going to be mostly alpha-1,4. It's kind of like glycogen. It's mostly alpha-1,4, some alpha-1,6. It's a little bit less. It's like 1 in 25. Glycogen is going to be about exactly 1 in 10. And cellulose is no branching. It's going to be, everything here is going to be alpha. Cellulose, though, is going to be beta-1,4. And this is why it's maybe difficult to digest cellulose. One, we lack an enzyme to help us digest it. But two, the beta-1,4 is going to be, remember, beta is stronger than alpha. So beta-1,4 is a stronger bond, so it's harder to break this down. I just also want to go on a note and say this is probably not super high yield, but could show up. So we're talking about it. There's a cool, fun picture. We have all alpha-1,4, mostly alpha-1,4, 1 in 25, alpha-1,6, mostly alpha-1,4, 1 in 10, alpha-1,6, and then all beta-1,4, very strong. Proteoglycans they're usually going to be extracellular matrix proteins or transmembrane proteins. You have this large core protein here, and the kind of branching off from it, you have these repeating disaccharide units, A, B, 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 A, B. And we call those glycosaminoglycans. Yeah, there's linker proteins and a little bit extra flair, but I think that's the main idea. It's this large protein with these disaccharide repeating units on the end. These are just examples of what the disaccharide repeating units could be, heparin sulfate. We're going to see others in a second. So again, you got this core protein, maybe a little bit of linker, a little bit of flair here, but tetrasaccharide, linker, blah, blah, blah. We're mostly interested in the A, B, 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 A, B. And it could be, there's patterns, etc., but I would just know that it's disaccharides. These are all the ones it could be. You could have hyaluronic acid, chondroitin sulfate, dermatan sulfate, heparin sulfate, keratin sulfate, etc. And these are just different types of, instead of A, B, this one might be A, C, etc. And this one might be D, C, etc. These are going to be really important. You can imagine a cell is like a fly stuck in a spider's web, right? And it's not the best example, but the spider's web is like anchoring it, giving it support, keeping it locked in. It's going to do more than just that, but that's always how I thought of it, anchoring it into place. We talked earlier about sorbitol and inositol being strong osmotic agents, being able to bring in a lot of water. So are these sugars, these glycosaminoglycans. There's lots of hydrogen bonding with the carbohydrate structure, and so we're going to pull in tons of water and hold it, sequester it. And so these are all these little water models attracted. And so my dad's a dermatologist, and a lot of people are asking about this, and I don't know exactly how it works. Some work better than others, but the idea is that if you can take this medication, you inject it, or you take pills, you're going to have more hyaluronic acid, more proteoglycans, and this can pull in water, and water in the skin is one of the things that gives it this nice, plump, youthful appearance. As you get older, you're going to lose a lot of your proteoglycans and glycosaminoglycans. I don't think it works perfectly though, because it's like, how do you get it exactly to where it needs to go, and how do you stabilize it? And things are much more complex than we're making it out to seem here. We can link sugars to proteins. You can do this to asparagine, and this is N-linked, and this is easy because it's asparagine with its amide structure here. Here's this nitrogen, and it happens in the endoplasmic reticulum. We then have serine, which can be O-linked. Remember that serine has an OH, and so we're seeing this, and this is going to happen in the Golgi apparatus. Every N-linked sugar is going to have the same pentasaccharide core, which we can then, so this is the mandatory. We'll always start with this, and then depending on the protein and the needs of the cell and the protein, we'll put other sugars, here, etc. The O-linked usually have the same core 2 or 3. For the N-linked, so again, we're going to have this core 1, 2, 3, 4, 5. And then we can, put other optional things on, and we're seeing more optional things being put on here and here as we process this. And this is, of course, happening in the endoplasmic reticulum for the most part. So it's going to start in the endoplasmic reticulum. We can certainly modify it in the Golgi, but the original asparagine pentasaccharide core is going to happen in endoplasmic reticulum. Important here is the idea that mannose can have its sixth carbon phosphorylated. So this is kind of a new point. So once we have the pentasaccharide on the N-linked asparagine, if you put a phosphate on a particular mannose on the sixth carbon, you have given it a postal code, an address that says this protein needs to go to the lysosome. If you remember from undergrad in the MCAT, the lysosome's job is it's kind of like a junkyard where we're gonna destroy old cars. We're gonna smash them up, and so old proteins are gonna be degraded in the lysosome. Remember, we had the alpha-2 macroglobulin earlier with Alzheimer's, and we said, okay, this job is to take the beta amyloid plaques from the extracellular position in Alzheimer's patients into the cells. And why is this good for helping us break them down? Because now we can put them in a lysosome, throw them in the wood chipper, and the lysosome hopefully will break these things up. So it has lots of acid, lots of hydrolases to break down. It's kind of like the stomach of the cell, right? It's going to break down a bunch of stuff, digest it. A lot of different uses, but definitely good for breaking down old, dysfunctional, misfolded proteins. But it only can do this if it has the enzymes necessary to break things down. And for those enzymes to get there, they need this phosphate on this mannose 6 position. Which leads us to eye cell disease, which I'll get into in a second. Let's just look at a picture here. We have an asparagine with this pentasaccharide core and then a particular mannose. We can go ahead and give it a phosphate on the 6th position. It will now interact with a receptor, and the receptor says, ding, 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 you are the right type of degradative enzyme. You should go to the lysosome, and it sends it there, and now it's here, and once it's there, it can do its job, right? Its job is just to sit here and to break stuff down, right? We have all of this stuff, and boom, now it's broken down because these degradative enzymes are doing their job because they got there because of the 6-phosphate mannose. All right, well, what is eye cell disease? Well, just kind of play out before I go to the next slide. What would happen if we couldn't phosphorylate this 6th position? What would happen? Well, the degradative enzymes, the hydrolases, wouldn't be able to get to work. They can't get to the lysosome. And if they can't get to the lysosome, what's going to happen? The cell doesn't know, it's not smart, it's going to keep sending things to the lysosome to be broken down, but they're unable to be broken down. That means the lysosomes will swell up with all of this garbage that they can't destroy. And also the enzymes that are supposed to be breaking things down, contained in the safety of the lysosome, are now out in the cell and out of the cell and in the plasma, and they might damage other things as well. And so this is eye cell disease. It might sound like a mild problem, but it's actually a very severe issue. It's really important for you to be able to break things down in your cells and your tissues when they're no longer necessary or they're dangerous. This is a cell filled with these large intracellular, these are all just giant lysosomes, which are just filled with garbage that we'll never be able to break down. And it causes just very severe skeletal malformations and developmental defects. All right, collagen. So there are 28 different types of collagens, 5 that you need to know. Honestly, most of them are type 1 though. So let's go through these. So type 1, and bun, 1, right? That's just how you should think of this. It's also technically in the skin and nails and eyes, but mostly the bone, I guess. So if you have a deficiency in collagen type 1, this is known as osteogenesis imperfecta. Well, we're thinking of bone and 1. So it makes sense that as somebody, there's technically a bunch of different types of osteogenesis imperfecta, but let's ignore that. Let's just say that you have a type 1 defect. You're going to have severe skeletal malformations because your bones are not functioning properly because they're missing this type 1 collagen. You're also going to have issues hearing. Why? Because the malleus, incus, and stapes are 3 bones in your middle ear, and they're going to help regulate the tympanic window, the tympanic membrane and the oval window, and just help modulate the hearing signal. So without these bones, with the type 1 collagen, you're not going to hear as well as you should. Your blue eyes, your sclera will become blue. Remember, this is also in the eyes a little bit. So blue eyes. So these are the 3 things. Your bones are messed up, you're having trouble hearing, and your eyes are going to be blue. That's type 1 collagen defect. That's osteogenesis imperfecta. Type 2, cartilage. Type 3, I remember that there's the intima, media, and adventitia of a blood vessel, and so there's this 3 layers, hence type 3. And there's Ehlers-Danlos is a disease we'll talk about, but one type is vascular Ehlers-Danlos, which will affect type 3. Type 4 for the floor, and the basement membrane is like the kind of the floor for a lot of tissue beds. Alport syndrome and the autoimmune Goodpasture syndrome are going to affect that. And then type 5 is skin and joints, and this is going to be the classic type of Ehlers-Danlos. And then we have a bunch of other types, but nobody cares. It doesn't matter. Collagen. So collagen is going to be this triplet repeat of amino acids. So 1, 2, 3, 1, 2, 3, 1, 2, 3. The first one always is a glycine, always having a glycine. And remember that if we're doing these little sharp turns, remember that glycine superlative has a really small R group. It just has a hydrogen. So it's able to make these turns really easily. So we have this 1, 2, 3, 1, 2, 3. And again, if you didn't have the glycine, there would be too much steric clash and you couldn't have such a tight turn. But the glycine enables that. 1, 2, 3, 1, 2, 3, 1, 2, 3. So this makes up a helix. And then 3 of these helixes, so this is now, let's call this A. I'm going to have A with B with C. 3 of these helices will wrap, and this is actually what we call a collagen molecule, is 3 of these smaller helices pushed together. And over here on the left, we just have there's lots of different types of collagen and makeups of collagen, and we have freaking 28 types, and it's probably even more complex than that. But It's almost, you're going to see glycine in every single one of them. So it's hugely abundant. Proline is very common as well. So I would know that it's mostly glycine and proline. You can have, of course, other things which we'll talk about. So let's go ahead and take a deeper look. So there's a couple different motifs. I wouldn't memorize these exactly, but it's often going to be glycine, some random amino acid, hydroxyproline. So in this first type of collagen, it would be glycine, some random one, hydroxyproline, glycine, some random hydroxyproline, glycine, some random hydroxyproline, etc. But it could also be glycine, proline, some random one, glycine, proline, some random one, glycine, proline, some random one, etc. It initially makes up a left-handed helix, and I always just thought that 3 lefts makes a right. And so remember that this is it's a triplet of triplets, right? It's a triplet in that it's 1, 2, 3, 1, 2, 3 with the amino acids, but then it's also, we're going to take our 3 left-handed helixes and put them together, and this makes a right-handed helix. This is actually what we now call a collagen molecule. We can now cross-link these together, and we get a collagen fiber. A lot of proline is quite abundant in collagen. We do have a little bit of lysine as well, but it's mostly proline. Proline can be hydroxylated. So here we have proline, and here we have our hydroxyl group put onto it. And the hydroxyl group is probably then going to grab onto a sugar. That's kind of the point of this. But this needs to be made, this is difficult. How do you just put an OH on something? You need an enzyme. This is a complicated thing. So we have proline hydroxylase to do this. But this tires the enzyme out, right? One important thing about enzymes is that they're supposed to be regenerated. This isn't fully regenerated on its own. It's tired. It needs to be revived. And what helps revive it, it's a little bit complex, but I would just know that vitamin C, ascorbic acid, is going to help revive the proline hydroxylase effectively so that it can continue working to hydroxylate prolines. Also, a different, lysine hydroxylase to, hydroxylate lysines. And this is necessary for hydrogen bonding and modifications and things like that. So that is why vitamin C is so important. And sailors, for much of, world history, we get scurvy because, you go out on a ship for 8 months and you're just eating fish and bread and you don't have any vitamin C. So you cannot hydroxylate your prolines and your lysines and your collagen. And we've seen collagen is in everything. It's in our joints, it's in our cartilage, and it's in our bone, it's in our skins, it's in our eyes. And that's why you get all of these classic symptoms of vitamin C deficiency, of scurvy. So just kind of an overview. We have these amino acid repeats, 1, 2, 3, 1, 2, 3, always with glycine is the first one. Often proline is somewhere in that triplet. We then take 3 of those left-handed helices, 3 lefts makes a right, and we make a right-handed supercoil. We now call that collagen. We are hydroxylating any proline and lysine, probably proline, but sometimes lysine. Now that we've hydroxylated them, we can glycosylate them. So you can add a glycosyl, you can add a sugar onto the proline or the lysine. We put 3 of those left-handed, we get a right-handed triple helix. We're then going to cut off the N and C domains from that triple helix, and then we're going to cross-link these things together. And it's just a very tough, very durable, slightly elastic structure. It's just really important, again, for so many different types of tissues. It's in the blood vessels, it's in the bones, it's in the cartilage, it is everywhere. Ehlers-Danlos occurs if, there's a lot of different types we'll kind of talk about, but main ones to know are the classical type, which is it'll say like col5A1, but I would just know it's collagen type 5. This is largely going to affect the skin and the joints, and people will be hyperflexible, hyperextensible skin. They're going to bruise easily. They might have something called like a Chiari malformation where their cerebellum is displaced a little bit. There's the hypermobile type, which will actually have a similar phenotype. And actually, this is an unknown gene. They don't actually know exactly how this one works. And then we'll have a vascular type, which is collagen type 3. Remember, 3 for the intima, the media, and the adventitia, also known as the externa. So collagen type 3 is the vascular type. This is affecting the blood vessels, can cause a mitral valve prolapse, an aortic dissection, aortic aneurysm, saccular aneurysm. You don't exactly know, you guys are just starting med school, so you don't really have to know exactly what these are, but it could show up on a test. So even if you don't know what a mitral valve prolapse is, just know that it's associated with type 3 Ehlers-Danlos, and as you go through med school, you'll pick up a lot of this stuff and you'll make connections. It'll be a little bit clearer. This is, you don't really need to know this, but just wanted to emphasize that there are more types of Ehlers-Danlos syndrome, right? There's so many steps in making the pre-collagen chain. We got to hydroxylate things, we got to glycosylate things, we've got to make sure we link up the 3 helices, we've got to cleave off the N and C terminus, we've then got to cross-link them. If you have defects anywhere in this process, you can get tons of different types of Ehlers-Danlos. But again, the ones we're mostly interested in are the vascular one, type 3 collagen, hypermobile, I think it's unknown, and then the classic one, which is going to be type 5 collagen. And that's it. I really hope that was helpful. Yeah, all the best to everybody.